Okay, well, we're just going to get started, and I'm sure some more people will trickle in. So wonderful to see everyone. Good evening, and thank you for tuning into our webinar, From Puzzle to Progress, Unlocking Rare Cancer Insights with Genomic Sequencing. We're the Rare Cancer Research Foundation, a nonprofit dedicated to curing rare cancers through strategic investments and innovative collaborations that facilitate effective research and accelerate the deployment of promising therapies. You'll hear more about us a little bit later, too, if you're not familiar. As a brief reminder before we get started, you are being recorded. You are also muted and your cameras are off so we can limit distractions and ensure a beneficial experience for everyone. But we do want to hear from you, so please feel free to leave your questions and comments in the chat on a rolling basis, and there will be time at the end of the presentation to address them. If for some reason there's a question that you ask tonight or you think of one later that is not addressed, please feel free to reach out to us at info@rarecancer.org. There will be a slide at the end of the presentation giving you that information, and we will do our best to get back to you in a timely manner. We want to make sure that this is an interactive experience and that we address all of your questions. So without further ado, I want to introduce our honored speaker tonight. Cody Barnett is a patient advocacy specialist with Illumina, which is a leading biotechnology company in San Diego that specializes in genomic analysis technologies. Cody is going to talk about the facts surrounding genomic sequencing and the empowerment it can provide for rare cancer patients and their providers to seek new and innovative treatment options. So thank you so much for joining us, Cody, and take it away.
Yeah, thank you, Emma. Hi, everyone, and welcome to today's webinar. I'm Cody. I work in patient advocacy at Illumina, where our mission is to improve human health by unlocking the power of the genome. And I'm really honored to be here with you to discuss genomic sequencing in rare cancers. Before I begin, I do need to acknowledge the following disclaimers. So I will not be providing medical advice or speak to individual cases today. Everything I share will be for informational purposes only. Always, always, always talk to your doctor if you have specific concerns. And then finally, Illumina's global patient advocacy team, we work with patients, families, carers, and the groups that represent them in order to build evidence and advocate for the positive impact of genomics utilization. One person's experience is not predictive of results in all cases, and your individual results may vary. So now that I've outlined all of those disclaimers, here's what we'll be discussing today. So we'll start with the basics about genetics. We'll discuss the role of molecular testing in cancer, and then we'll focus specifically on the use of whole genome and whole transcriptome sequencing in rare cancers. We'll end by looking at what access looks like here in the US, and then Barbara and I are going to have a little bit of a discussion and hopefully answer some of your questions. And if you don't know what genomics or transcriptomics mean yet, don't worry, we're going to go over that and really try to make that as simple as possible. So to get started, we're just going to, again, look at those building blocks, and this will be the foundation for the rest of our discussion. Okay, so DNA stands for deoxyribonucleic acid, and it's the instruction manual for life. And it's found in cells throughout the body. DNA tells each cell how to grow, function, and respond to its environment. It's what makes you, you. And genes are made of sections of DNA that carry specific instructions for proteins. And genes shape our traits, our health, and how our bodies react to disease. Genetics is the study of how changes or mutations in genes affect how our bodies work. And humans have about 20,000 genes in our body. And the genome is the full set of DNA, including all of the genes. And the human genome has 3.2 billion with the B base pairs. And to put that into perspective, the National Human Genome Research Initiative said that one single human genome produces 200 gigabytes of data. And that's the equivalent of maybe the movie Jaws, you know, the shark movie from 1975. that's the equivalent of 200 copies of that film. And that's all in one person's genome. And genomics is the study of the entire genome of a person. And it helps us understand how DNA affects health, traits, and disease in a really comprehensive way. And I also always like to say that the genomics is a new field. And in many ways, it's incomplete. And that's because we are still learning so much about what the genome is and how we can interpret it. So what we know today is a lot more than we knew 5 years ago, but it's really small in comparison to what we're going to know 5 years from now. So it really is an incomplete science. And part of this conversation is really kind of understanding where we are in the context of genomics today. And then what I really want to emphasize is that when you look at a gene, you get a puzzle piece. And when you look at a genome, you get the puzzle. Well, the puzzle piece can give you important information. You have to know exactly where to look. And while the puzzle clearly has the full picture, you now need to put it together to really make sense of it. And we're going to go back and forth between these throughout our conversation today. And just keep thinking about the puzzle and then versus the whole puzzle pieces that we still have to put together. And cancer is a disease of the genome, and it all starts with DNA. So when your cells divide, which they're doing constantly, your DNA is copied, and sometimes mistakes in this process are made, and these are called mutations. Mutated DNA can give cell faulty instructions, leading it to behave differently. So when we talk about mutations, we're in the context of cancer, we're typically looking at oncogenes, which push cells to grow fast, too fast. We also have tumor suppressor genes, which can be turned off, that normally operate to keep that growth in check. And then we also have damage to the DNA repair genes, which make fixing mistakes harder in the future. So you just accumulate more of those over time. And cancer occurs when these dysregulated cells proliferate and then take over. So the definition of cancer is unregulated cell growth, and that's really what we're talking about. So if you think of the genome as a cookbook, and each cell is preparing a recipe, it's the DNA that provides the step-by-step directions. And mutations can be small, like a misspelled ingredient. And in that case, it probably doesn't matter at all. Or they can be significant, like an entire page that was printed back to front. And in that case, we can start to see why this would be really problematic. Okay, so this first graphic on the left side shows what we call a proto-oncogene. So proto really just means normal, how it operates to promote growth. And then it's balanced by a normal tumor suppressor gene that is suppressing growth. So as we can see here, everything is in equilibrium. And that's really what our body is like. That's what we want to see. But in cancer, the proto-oncogene becomes mutated. So now it is an oncogene, and it has what we call a gain of function. So it is promoting growth, but in an uncontrolled way. And then when we have a mutated tumor suppressor gene, we have a loss of function. So that means it's no longer doing its job. So it's not suppressing that growth at all. And that's when we start to see the uncontrolled growth that's indicative of cancer. And this is just another way of looking at that same information. We really like to see in healthy cells an equilibrium. So the proto-oncogene is pushing on the gas. Well, the tumor suppressor gene is putting on the brake, so we stand still. And when we have mutations that are leading to cancer, that means that either the proto-oncogene is pushing on the gas too far and the tumor suppressor genes just can't keep up, or the inverse, where the tumor suppressor genes become inactivated, meaning there is no brake at all. And now we just have that growth that we've already talked about. And then in the context of cancer mutations, we have two broad categories. So we have what are called germline DNA changes, which are present in every single cell of the body. They are, you have them at birth. So these are things that you inherited from your genetic parents. And they can have treatment implications. And these are probably most commonly understood as like the BRCA1 or the BRCA2 variants that can increase someone's likelihood of developing things like breast or ovarian cancer. Um, and it's also important to mention that these germline DNA changes don't cause cancer, but they do increase your risk of acquiring cancer. And then we have what are called somatic DNA changes, and these are the ones that are acquired during life. And a lot of these are just a natural part of aging, and that's why we tend to see more cancers in people as they get older. But they also can come about when we pressure test our systems. And you could do this by like smoking or being outside in the sun a lot. And that's why we see skin cancers more in people who have those behaviors. But it also can just be breathing the air in an environment that has a lot of pollutants in it. And these sort of mutations are not in all cells of the body. They're really only found in the tumor tissue itself. And they cannot be passed to children. So that's an important note. So these are not things that you can pass along to your family. So now that we have a little bit of an understanding of how genes work and how mutations can drive cancer, let's look at how molecular testing can help us detect those changes, and then how we can use that information to guide diagnosis, treatment, and research. So a biomarker is something in your body that can be measured to give clues about your health. And you probably are really familiar with them from all different instances about your life. So it could be something as simple as your temperature or your blood pressure or even your LDL cholesterol level. It really is just something that you can measure that gives insight into health. But in cancer, when we use the word biomarker, we're actually talking about the gene mutations that are actually helping your cancer to grow or survive. And testing for biomarkers is really important because it gives doctors a lot more information about the specific type of cancer that you have. And it can guide decisions about what might be the best therapy options for you. I also always like to mention that there's a lot of names for this, and none of them are wrong. It's just depending on where you are in the country or what resource you're reading. So you might also see this as genomic testing, tumor profiling, molecular testing, next generation sequencing. The list can go on, but just know that we are all talking about the same thing with those terms. And the reason why biomarker testing is so important is because we've learned that when people have a biomarker that's identified, so a mutation that's identified, that we can then match to a therapy, their health improves. So their overall survival is much higher. And we see this across the board where it is an option. And for this slide, I really just like to talk about before the blue area, what we call next generation sequencing, it really wasn't feasible to develop drugs for rare genomic alterations because each and every mutation required a specific test in order to find it. So choosing the right test for a mutation that only 1 in 1,000 people was like searching for a needle in a haystack, and it really just wasn't practical. But once we started to have the ability for next generation sequencing, we can now check for hundreds of mutations at once. And that's really what made small panels and then these larger panels that we were talking about. So these are looking at over 300 genes, and this is really standard of care for most cancers. But we're now on the cusp of really looking at how we can apply whole genome or whole exome sequencing to cancer. And that's really going beyond what's in a pre-selected panel to look at the entire genome for insight into how that could factor into your journey and treatment planning. Okay, so mapping the first human genome was a really big endeavor. So the Human Genome Project took over 13 years, over $3 billion, and the combined brain power of 20 universities across 6 countries. Today, we can map an entire genome for less than $200 in less than 24 hours. And whole genome sequencing is used to read all of the DNA of an organism, and it finds the orders of every single letter of the DNA one by one. And in people, remember, that's 3.2 billion base pairs. So that's a lot of information. Uh, Whole exome sequencing is similar, but instead of looking at the entire genome, it's really restricting its review to only the part of it that's responsible for protein. So it's about 2% of the total genome, but still a lot more information than what we get from traditional biomarker testing. And whole genome sequencing is really widely used to diagnose genetic or rare diseases and is at the heart of many of the gene therapies that you may have read about or seen on TV. And it's just within the last decade or more that we've had the clarity of understanding what is happening at the genomic level in order to actually make some of those research advances. Wow, excuse me. Okay, so I'm with you. If it wasn't already complicated enough, we often then combine whole genome sequencing with what's called whole transcriptome sequencing. So whole genome looks at the DNA and whole transcriptome looks at the RNA. And because transcriptomics doesn't roll off the tongue, we also often call this RNA-seq. And if whole genome sequencing helps us see the entire movie library on a streaming service, you can see everything that's there. Whole transcriptome sequencing shows us what someone's actually streaming. And it shows us how often they're streaming it and where. And that's really the advantage of combining the two. So taken together, it shows us not only what mutations are present, but how they're actually active, how they're actually acting on a tumor at a molecular level. And at Illumina, we talk a lot about how whole genome, whole transcriptome sequencing is really a universal test when it comes to cancer, because it gives us insight into those germline changes, so things that you're born with. So not only your cancer risk, but also how your individual body is likely to respond to any medicine. So we call that pharmacogenomics. And then it also looks at the tumor-specific features that we know are really important for understanding what the actual diagnosis is, what the prognosis is, what are the likely treatment options that are likely to work. And then it also gives us insight into is someone responding to therapy and what are their risks for relapse. And when we talk about the clinical benefits of whole genome transcriptome sequencing, and to be honest, they're not all that different from traditional biomarker testing. It's just the volume of information that we have available. So whole genome transcriptome sequencing can be really helpful in improving or refining a diagnosis. And this is really important when we're talking about a rare cancer where maybe a pathologist isn't used to seeing it very regularly. I was talking with a patient advocate leader a couple of days ago, and there's over 100 different types of sarcoma, and that's a lot for any one person to manage. And that's where things like whole genome sequencing can really be helpful. It also can help inform treatment selection, and then also clinical trial eligibility. So by understanding the mutations that are present, it gives us an unparalleled view into what are the possible therapeutic options and what those might be for the tumor and patient in front of you. And then it also gives really great insight into clinical trial eligibility. And this is really important because today over 70% of clinical trials do have some sort of a mutation eligibility criteria that they're looking for. And then it also helps us understand resistance if it's forming. So this is for someone who, maybe they've responded very well to a therapy, but now are no longer responding. And the whole genome sequencing can really help us see at a molecular level how the tumor has responded to that therapy to kind of overcome it. And that can be really important to plan next steps. And then because again, whole genome is looking at those germline factors, it also gives us information about the inherited risk for family. And the more that we're doing this sort of testing, the more that we're starting to understand that it's not as simple as we once thought it to be. So some cancers, I worked in melanoma for a really long time. And we used to always say that melanoma wasn't something that is inherited. It's because families have shared risk behaviors. But we now understand that it's not that simple. And about 10% of melanomas are in fact inheritable, or the risk or increased risk for melanomas are inheritable. So this is really important insight that whole genome transcriptome sequencing can provide. And I'm not going to go through all of this just because I think we have a lot to go over. But this really isn't that different than traditional biomarker testing, or at least the sequence of events. But where it is different is this data interpretation piece. So because we have so much more information that whole genome sequencing is looking at compared to biomarker testing, there just is a lot more data to interpret. And then because of that, you really want to make sure that you have a team who's prepared to do that analysis in a way that really gets you the value of having done the test. Okay, and there is a lot of words on this slide, and I apologize for that. But what I really want to point out is that biomarker testing, because we're looking at a pre-selected set of what are called clinically relevant genes, you're really limited to that. So some biomarker or comprehensive tests are looking at 300 genes. A lot of them now are at 500 or a little over 500 genes. And while that's great, if your cancer has a mutation that's within that group, it doesn't provide any information if your mutation isn't part of that group. So you could have biomarker testing done and get a report that says that you have no findings. And it doesn't mean that there isn't a mutation. It just means that the test that we're using isn't designed to pick it up. And that's the benefit of whole genome sequencing in comparison. So biomarker testing is really designed to match patients to approved targeted therapies or immunotherapies, where whole genome sequencing is more exploratory, and it helps discover novel or rare mutations that are present in a cancer or in a tumor. And that really can be important when we're talking about a rare cancer. Okay, so I really like this slide because I just think that it visualizes a lot of what we're just saying. So if standard biomarker testing is like watching a trailer of a movie, you get all of the highlights, but you miss all of the detail. Whereas whole genome sequencing is like watching the full film all 200 times. So if you go back to that Jaws analogy that we used earlier, that's a lot of shark. And you can see how you can get a lot more information when you're using a test that is looking for that extra information. Okay, so now we're going to pivot and look specifically about rare cancers. And this slide you've already seen. It's the exact same text, so no need to look at it super carefully. But I do really want to hone in on these first three areas. So for diagnosis, we know that up to 30% of rare cancers are misclassified or misdiagnosed. We also know that many cancers are diagnosed via pathological examination. So that means at the end of the day, it's a visual test that's being used. And if a pathologist has never seen your cancer or has seen it infrequently, it can be really difficult to pick up one rare cancer or one subtype of the same sarcoma, for example, from another. And we also know that rare cancer patients are bounced around between different specialists, and it can be frustrating, and just as importantly, it can take a lot of time, and time is really important. So whole genome sequencing can dramatically shorten the period it takes to actually get to that accurate diagnosis. And then for treatment selection, we also know that whole genome sequencing can detect rare or novel mutations, including structural variants and gene fusions that, that a targeted panel or biomarker testing might miss. And that can, again, give you really comprehensive look at what all of your options might be. And then for clinical trials, again, this is just really important in understanding all of the options that you have available for you. So I now want to switch to just a couple of studies that point out some of the data that backs up some of the things that I've been talking to you about. So in this study, looking at 200 soft tissue and bone tumor sarcomas that were suspected to be, I'm sorry, I guess they weren't sarcomas yet. So they were 200 soft tissue or bone tumors that they thought were malignant. And then they tested them using whole genome or transcriptome sequencing. And what they found is that whole genome transcriptome sequencing changed the diagnosis in 13 out of 197 cases, so about 7% of the time. So 4 of those cases were initially believed to be sarcoma. They were actually benign, which is great news for that patient or for those patients. And 2 were actually metastatic melanomas, which have a very different treatment plan than sarcoma. So that shows how genomic data can really help correct misclassification of tumors. And then it also found that whole genome sequencing with transcriptome sequencing found that 15% of cases, they found treatment relevant variants or mutations, which made those folks opened up the doors for those folks to receive targeted therapy or to enter into specific clinical trials. And I think one of the most interesting things in this study is that 11% of the participants carried germline variants or mutations that made them at increased risk for developing those cancers, which is really important to know for the rest of the family. Okay, so I'm going to skip this one just because this is for a leukemia. It's not as rare of a cancer, and I'm also just worried I'm running out of time here. But this next one is a really interesting study. So it's looking at over 1,300 patients, 3/4 of which had rare cancers. So 75% had rare cancers. And whole genome transcriptome sequencing offered therapy recommendations in 88% of cases. So almost 9 out of 10 cases got a recommendation due to sequencing. And then nearly 1 in 3 patients received a matched therapy based on the findings. So that meant that of that 90%, 1 in 3 of them went on to actually get the therapy that was recommended by testing. And then of that group, a little over 1/3, so 35%, achieved progression-free survival at a much higher rate than the other folks involved in the study. And what that really shows is that whole genome transcriptome sequencing provides actionable insights for rare cancers where standard options might be limited. And just to kind of reiterate, the benefits to rare cancers really are a faster, more accurate diagnosis. We know that rare cancers can be hard to identify, and whole genome transcriptome sequencing can reduce delays and help doctors reach the right diagnosis sooner. We also know that it helps find hidden genetic clues. And that's just because whole genome transcriptome sequencing is detecting a huge wide range of genetic changes and not just that pre-selected panel that standard biomarker testing is looking at. And then we also know that it can support personalized care in a way that it is really truly personalized because we're looking at all of your genomic information. And then the last point that I think Barbara and I will definitely talk about later is just that when you have this genomic sequencing data, it can be really helpful in accelerating research and contributing that data to researchers in a way that they can use it can be really important, particularly in rare cancers. Okay, so now I have a patient example. So this is Balwinder. He is 58 years old. And 3 years ago, so in 2022, he was diagnosed with an aggressive sarcoma in September. And they tried all of the standard treatment options. None of them worked. And in November, so 2 months later, he applied for and got permission for whole genome sequencing, and it revealed a genetic signature that opens the doors to him accessing immunotherapy using a compassionate use program. Um, Within weeks, he was showing signs of significant recovery. He regained mobility, independence, and is doing quite well today. And that's because the whole genome sequencing created a personalized, tailored treatment approach that would not have been available to him without it. Okay, so this is the final section. We're gonna go through this pretty quickly and get to some of your questions and discussion. When we think about access in the US, I wanna be upfront that it is limited. There are only a very small fraction of patients have access to this right now. However, it is growing. There are concerns about costs and reimbursement at the systems level. Clinical guidelines often do not include this, although some are starting to. There are concerns about actionability. So when you look at the entire genome instead of those 300 genes in biomarker testing, you might get a result that is interesting from a research perspective, but there might not be something that we can do clinically for it. And that's something that is a concern. However, I would say that I would rather know and know what that mutation is. Maybe I could channel my advocacy towards research on that specific mutation. But it is something that we need to acknowledge is a concern. And then it's also important to recognize that there are real infrastructure or capacity concerns with this sort of testing. Not everywhere can do it. It does require specialized equipment and specialized training, but it is happening. So at Memorial Sloan Kettering, Dana-Farber, St. Jude's, in fact, many children's hospitals are regularly using whole genome transcriptome sequencing. And then globally, we're seeing the United Kingdom is really leading the way. But we also have initiatives in Germany, Sweden, the Netherlands, Japan. And that's because people are really starting to see the relative benefits of this over biomarkers. And we're also seeing that awareness is growing. And as awareness grows, access will too. So if you're interested in this, I suggest really just talking to your doctor about it. Like I said, this is not something that all doctors are comfortable with. I know Barbara has a lot of experience in kind of interfacing with clinicians and really helping them understand. We're going to get to that as part of our discussion. But asking for this in the context of, could this provide additional clarity into my diagnosis can be really helpful to ask. So I want to thank you all for your attention. And now I think Barbara is going to join, and we're just going to have a little bit of a discussion and then answer your questions. So thank you again. And Barbara, you are muted.
Hello, everyone. Can you hear me now?
You can.
Okay, I'm Barbara Van Hare, and I am head of partnerships at the Rare Cancer Research Foundation, and I also manage the day-to-day biologistics for Pattern.org. Just for a little background, for those of you that may not know what Pattern.org is, it's an initiative of RCRF that allows patients, regardless of where they're being treated across the US, whether it's a large academic center or a small community hospital, they can give excess tissue from surgeries or fluid from fluid drains or even stored tissue and or clinical medical data to research, again, regardless of where they're being treated in the US. And so we thought it might be helpful if Cody and I asked each other some questions just to help make this whole idea of sequencing a little more personal. So Cody, I'll ask you a question. You mentioned during your presentation that genomics is an incomplete science. Can you explain a little more what you mean by that?
Yeah, so when we talk about genomics being an incomplete science, what we're actually saying is, um, and this is true of all sciences, but it's particularly true of genomics, that we just acknowledge right up front that we don't have all of the answers yet. So when I work with folks on the rare disease side, we might identify mutations, but not actually know if they're causing the condition in front of us. And that's just because we don't know enough about all of the genes in the body to actually say what's doing what. And that's also true in this context as well. So I think when we talk about it being an incomplete science, it's really just us trying to be upfront about the fact that there are limitations here. This isn't a yes, no kind of a test, and it really requires interpretation. And we might not have all of the information we need to interpret things correctly quite yet.
Great, thank you.
And Barbara, I also want to ask you, like, I know that you've had some personal experience with this. Would you mind sharing about that?
Sure, I'm happy to. So back when I was young, 2 years old, I have a family history of different rare cancers. When I was 2 years old, my father was diagnosed with mouth cancer. My brother in 2009 was diagnosed with GBM, which is the deadliest brain cancer there is. He's still alive today, thank goodness. He's a walking miracle. In 2012, I had a nephew that was diagnosed with ocular melanoma. And in 2019, I think it was, I was diagnosed with a more common cancer called melanoma. Unfortunately, my nephew's ocular melanoma diagnosis, which is eye cancer, metastasized in late 2020. And through his journey as the cancer spread throughout his body, he would have there were different tumors in his body, and they would be sent off to have whole exome sequencing done. And he had had several tumors sent to different places to study, and it would show different mutations in the tumors. But the first tests that were done at different companies did not show in the report any germline mutation, meaning that there was some family history. And we figured somewhere, because of all of these cancers, there must be something somewhere. But as Cody said, not everything is known yet. Well, then a lesion, a tumor piece was actually sent to one company. It just happened to be Boston Gene up in Cambridge, Mass. And on that report came back that actually he did have a germline mutation, meaning he'd inherited something through the family.
Wow.
So, being the type of inquisitive person he was, he got a bioinformatician to look at the data from where they'd been sent before, and actually that germline mutation was in the data, but because his germline mutation was not very well known, very few people in the world today know of it. A few do. It's called a POT1 mutation, which is basically it's a predisposition syndrome, meaning you're more likely to have, I think there's 6 or 7 cancers, none of which you really want to have. Um, and so I ended up, after I had a second melanoma, I actually got sequencing done, and it turns out that I have that same mutation. The good news, bad news is I have that mutation, so I know that I'm predisposed to have one of several cancers. I have now had 5 melanomas, but they've all been stage 0. Because I am aware of this risk, I'm able to be monitored every 3 or 4 months to see a dermatologist with specialized photography that maps every mole on my body and looks for changes. And they've also found that I have the beginnings of a blood cancer called CLL. They have no idea if I've had it for 2 years or if I've had it for 50 years.
Wow.
But again, it all is tied back to this genetic mutation. If the sequencing had not been done for my nephew, I would not know today. I wouldn't be able to be proactive. I wouldn't be checked every 3 to 4 months as I am now. So literally, I feel that the power of sequencing will probably or has already saved my life because I know what to look for, my doctors know what to look for, and they're still learning. Again, as you said, it's we don't know everything yet. I personally feel there are probably a lot more pot 1 patients out there, but most of the tests that don't test the whole genome or the whole exome don't pick up on that. So I think that's just one example that there's a lot we don't know. And so sequencing can really help researchers learn what is helping drive some of these cancers and help people find out if they do have a risk. And so hopefully it will save millions of lives, over time.
Wow. Thank you for sharing, Barbara. I
Welcome.
really appreciate that. I'm also curious, like I know the Rare Cancer Research Fund is really invested in this idea and then also using the data to help accelerate science. Could you talk a little bit about that?
Sure. I have a phrase that I have a tendency to say in that, and it's true, patients have what researchers need. And you say, well, what could that be? Patients are the ones that grow the tissue. They're the ones that have the tumors. That tissue is very important to science in order to get it to move forward. The data is as well. So tissue and data, what researchers are actually doing, whether it's tissue, whether it's data, whether it's both, is they are looking for patterns in that tissue and data. And so whole exome and whole genome sequencing are just providing lots of data that are going to help researchers get to answers that are needed for patients in finding new therapeutic options for rare cancers, for common cancers, even for rare diseases, and even some common diseases. So with patterns, since we're dealing with patients that are donating tissue and data, um, anybody that signs up through pattern.org to donate any of those, we can work with them and do work with them to see if they're interested in getting whole genome or whole exome sequencing. In many cases, patients have never heard of it, so we can help walk them through what it is and how it can be helpful. Either they can go back and talk to their doctor, or we're willing to talk to their doctor with them or for them to see if the doctor, because to get these tests from a clinical use, in order for the results to come back to the clinician to help the patient, the clinician will need to sign off. And unfortunately, there are a lot of doctors that don't really understand sequencing, but once you explain that to them, in most cases, they're very willing to sign. And so we can help kind of facilitate that discussion and that process, and even put the patients in touch with one of many companies that do the sequencing, or if the doctor has a preference, to help them get the test done and also know the cost up front if there is going to be any cost for them. In most cases, insurance will cover it. It kind of depends. If
Yeah,
insurance doesn't, many of the companies have funding that can help bring down that cost, but the patient could know that up front.
yeah, and that's really important to know. Um, I'm also thinking, sorry, looking at some of the questions that are coming in. Um, And I'm sorry, some of these I don't think we're going to be able to answer just because I think that they're a little too specific on kind of someone's like medical care. But I think we can address, someone asked about what are the types of, sorry, if there is a comprehensive list of genomic testing. And what I think this person might be asking is, is there a list of testing companies? And I don't have that list. Barbara, I know you have a list of companies that you've worked with before that I'm sure you can help people with. Um,
Yes, we can work with any of the companies. Some of the better known names, I'm sure there are a lot that we don't know, are like Caris, Tempus, Boston Gene. Um, I don't know all the names of the different ones, but if you're interested, they can reach out to us at info@rarecancer.org and we can get back to them. Many doctors have the companies that are kind of their favorite to work with or their institutions work with. Um,
And then someone has asked, how does someone go about donating tissue to the Rare Cancer Research Foundation?
If someone is interested, they can go directly to pattern.org. And in case you hadn't guessed, the reason why it's called pattern.org is because the real goal is for researchers to find patterns in that tissue or data that the person donates. If a person goes to pattern.org, they can read about the process. They put in some basic information about themselves and their particular diagnosis, and then they are matched to a study if they have one available. We also have a biobank for some cancers if there's not an actual study active yet, and we'll either work to find one or know that one is coming up in the fairly near future. And they can go ahead and consent online with pattern.org. Now they can consent if they have a surgery coming up or a fluid drain. They can sign up. If they have stored tissue they'd like to donate, they can sign up that way. And if they don't have any tissue, either because they don't have a surgery or they don't have any stored or they don't want to donate tissue, that is fine. If they want, they can donate their medical information, their medical records. We'll do all the legwork to pull those. And if someone has had sequencing done or plans to get it, if they're interested, they can donate that raw data. We have a form that we can send them, and we can put that in what we call the pattern data commons, which would all be de-identified. And so researchers can look at all of that data. Our goal is to get as much data in there. So again, researchers and bioinformaticians can find new learnings that will save lives,
Yeah,
prolong or save lives.
that's really great. And it's nice how easy you all make it. I am seeing a question about whether or not this would pick up mutations in CSF for leptomeningeal disease. And when we're doing whole genome transcriptome sequencing in for cancer, the way that they actually do it is they'll look at either a sample of tumor, if it's a solid tumor, or they'll do a liquid biopsy and actually get some of the cells from the tumor. And then they'll actually look at like a healthy part, like a healthy cell, and we'll look at the genome from the person. And then they basically subtract one from the other so that all that's left is the genome of the tumor. And that really does give them a lot of insight, regardless of where it is in the body. So I don't know specifically for leptomeningeal disease.
I can answer that actually, Cody.
Yeah, that's great.
We've actually gotten CS fluid from several patients and sent it off for sequencing. So it is something that we can do. It's actually a fairly easy procedure because the fluid is coming off and we can send a kit and it can be collected.
Amazing.
We do have a question, it looks like from Michelle Carter. She wants to know if we can talk to the frequency in which someone in her situation should have testing. She says her disease is highly metastatic. Um, Her observation is that many doctors don't proactively suggest testing, which is something we've seen in our experience as well. And yeah, she says it's important to start this conversation with your doctor. So if you guys have any input on that, I agree, Michelle. That's a really great insight.
Would you like me to handle that one, Cody, or
Do
would
you want to kick us off and I'll add?
Okay, that would be great. I think that that is a discussion to be had with your individual clinician or clinicians. I think you're right. Many times they don't want to continually request sequencing, but one of the things we do know about a lot of cancers is um, a cancer may show a certain thing in one tumor, but as the tumor mutates and more tumors come about, different mutations can be shown as more tumors. And so I think it's working with your clinician to see what they can do or working with a group that can help try to convince the doctor to get more testing. In the case of my nephew, he was probably his own best advocate with convincing doctors because not only was he working to kind of help his disease, he was freely sharing his data that hopefully would help the researchers. And that seemed to help more doctors be willing to order the test in his case. Cody,
Yeah.
would you like to add to
And
that?
that's great, Barbara. I agree. This really is a great question for you and your care team. That said, we see a lot of utility for whole genome, whole transcriptome sequencing at diagnosis. And while you could make the case for additional sequencing at recurrence, like what we've discussed already, this might actually be an area where a liquid biopsy, which looks at circulating tumor DNA, might be ideal. With a liquid biopsy, your doctors would take a blood sample and then sequence that and compare the findings against the baseline you already had through whole genome, whole transcriptome sequencing. This can detect new mutations, resistance mechanisms, and tumor heterogeneity that may have emerged since your initial treatment. Liquid biopsy is also faster, and because looking at circulating tumor DNA found, and by looking at circulating tumor DNA found in blood, it can also pick up if tumors in different parts of the body have different mutations, which sometimes can happen. And this really is a great use of liquid biopsy as a technology. And to your point about doctors not always being proactive about testing, I agree. And this is something to really keep in mind is that this is new for them too. So you asking, you bringing it up at your appointment, that can be really helpful for them as a trigger, showing that you're interested in knowing everything possible about your cancer, about your tumor.
Absolutely. I have a few other questions, and everyone, please feel free to continue submitting your questions. We'll get to them as soon as we can. But I had a few pre-submitted questions that I think are really important to address for everyone. So what is the average cost for a patient to get genomic sequencing? We talk about access, and cost is
Yeah,
something to consider.
that's a great question. And it's one that I wish I had a more simple answer. So the way our healthcare system works is every insurer is going to have kind of partnering labs. They're going to have negotiated rates. Everyone has a copay or a deductible, and it depends on where they are in the kind of that cycle. So it's really difficult for us to give kind of this is what it costs because there's so many variables that go into it. But what I will say is that there are often programs that do exist at labs that do make it affordable. They can also test and reach out to your insurance company for you to see if it's going to be covered and what your out-of-pocket costs will be before it happens. And they're very happy to do that. Barbara, I know you've done this a lot. So what is your experience?
I would say overall that's very true. Usually they're very happy to do that. Again, for patients that are, donating through us, we're more than happy to try to, if there are any sticking points, to get involved to try to make it even easier for the patient.
Absolutely. Great. So zooming out from an individual lens to a broader perspective, how might someone's genomic sequencing data help other patients with that same cancer type, especially if they choose to donate that data through a program like ours?
Yeah, and I think this is a really fascinating time when we talk about data because nowhere else in history has it worked quite the same way. Where when you're doing even biomarker testing and you're contributing that data, not only is it helping you in that moment because it is helping to inform your treatment options, it's giving your provider really important information about prognosis and what's actually happening at a molecular level. So not only are you getting benefit, but that data is also really important for researchers or for pharmaceutical companies who are looking at how do they design therapies that are meeting unmet medical need. And when you contribute your data is now being counted, and they're now considering that in their work as they develop new and potentially better therapies than what are available today.
Right. Yeah, absolutely. So, referring to your presentation, can you explain more about how whole genome sequencing helped the patient that you mentioned get immunotherapy? And, maybe for some people in the audience, can you explain what immunotherapy is in basic terms?
Yeah, yeah. So in this particular case, he actually lives in the UK, and they have their healthcare system is different, and to demonstrate that immunotherapy would work, to become eligible for immunotherapy through a compassionate use program, they had to have some semblance of like a real hypothesis about why it might work. And what the genomic testing was able to give was to demonstrate that there was a reason why the doctors wanted to try this. And immunotherapy is different from other forms of other therapies in that it doesn't work on the tumor directly. It actually works on the immune system to help the immune system to identify and then attack those cancerous cells. So it's not a chemotherapy, it's not a targeted therapy. It really is acting on the immune system itself, kind of releasing those brakes is what we used to describe when I worked in melanoma, so that the immune system can identify and attack those cancerous cells. Um, so that's one of those use cases of whole genome sequencing, is that because it's hypothesis neutral, you're not going in there with any preconceived notion about what you're going to find. You're going to look at all of the data, and then you're going to see and interpret what might be of use. That's really the use case for this, because in this specific situation, it's not something they would have looked for otherwise.
Right, absolutely. Absolutely. Barbara had mentioned this a little bit earlier. This is my last pre-submitted question. So again, if anyone wants to submit any questions, we would love to have them. Can Illumina or the Rare Cancer Research Foundation help me get access to whole genome sequencing if my doctor doesn't know what it is or how to request it?
Would you like me to answer that, Cody,
I
or?
think this one's all you, Barbara.
Okay. Okay. Yes, we're happy to try to help patients get access to it. I'll just give another story from a patient that had reached out to us, was willing to donate part of their sample to through pattern.org to research. And we asked, did she know about sequencing? And she did not. Most cancer patients are pretty overwhelmed with their diagnosis when they're first diagnosed and directly after surgery. So she didn't ask a lot of questions. She had a few, and we were able to answer them. And she said, "Well, okay, I'm going to go back and talk to my doctor," which she did. A medical oncologist in a fairly large city, um, who, quite frankly, had never heard of sequencing or what it was. But to his credit, he said, I don't know much about it. I don't really know anything about it, but if they think it might help, then I'm glad to sign the forms. So we sent the forms, he signed, the patient signed, they got their sequencing. The doctor were able to help coordinate so the doctor understood the report. The reports that they usually send are fairly easy to understand, but if they have questions, a lot of these companies have people they can ask. And then the patient went on to get treatment based on what the report had indicated, what was needed. And the particular patient I'm talking about was diagnosed, I think, 2 years ago, and is doing fairly well, actually doing very well at this point.
That's great news. That's great. So again, if anyone has any questions, please feel free to let us know. Cody's going to pop up a slide here for me, and it'll just remind you what our primary email address is for RCRF for your questions, comments, concerns. So yes, thank you so much for tuning in this evening. Um, as I said, if a question comes to you later, feel free to send us an email. We're happy to help however we can. And those are our social media channels. So please feel free to give us a follow there and interact with our content. And the webinar recording will be available next week. So if there's no other questions or comments, again, thank you so much for watching. Thank you, Cody, for a great presentation, and Barbara, and for your interaction. And yeah, we hope everyone has a great evening.
Thanks, everybody.
Thank you. Thank you, Cody.